Hello and welcome back. In this video, we're going to cover part 2 of the concepts, the scientific concepts that you'll often see in the ACT science section. Again, if you haven't seen part 1, go watch that. And as I said there and in my other videos, the ACT science section, despite the name, isn't really about science concepts. It's mostly about reading graphs and tables and knowing how to find information. So if you haven't seen the other videos on the introduction and the graph types and the question types, go watch those first, because that will cover the vast majority of the section. The science concept videos are what you're going to go to next, because they will cover the few scientific concepts that you do need to know. There's only going to be a few questions, or they might help you just better understand certain passages and avoid confusion. So here in part 2, we're going to cover some of the more complicated concepts that a lot of students haven't run into, or they have, but they just need a refresher on, but they're still commonly seen. Especially in this case, there's often going to be a passage based around them, especially if we're talking about the voltage, magnetism, that sort of thing. Almost no students have run into that before the ACT. You're not expected to. And that's why they're normally going to be the last passage or two, because they're meant to be hard. Now, you don't really need to know this much stuff for them, but it helps to know just a few little things. But as always, if you find this helpful, you can help me out by leaving a like and subscribing. Let's get started. So we're going to begin with genetics. This is actually one that a lot of students, depending on when they're starting to study for their ACT, have learned in their biology courses. But it's extremely common to have a passage based on genetics, and this is probably the one type of passage where it actually is really critical to understand something and how genetics work. So let's cover that. And we're going to start with an example here that I just made up, where we've got some dogs, where we're going to have a couple traits, that being the dog's fur color, which is going to be determined by the A genes, and the dog's eye color, which is the B genes. So for the fur, capital A will give us tan, lowercase a will give us black. For the eye colors, capital B will give us brown eyes, lowercase b will give us blue eyes. And so for my examples, I'm going to have two dogs. So here for dog 1, we're going to have capital A little a, and then capital B little b, so tan fur, brown eyes. And then dog 2, I'll have capital A little a again, so again tan fur, and little b, so blue eyes. And I'll cover a little bit more where I have it written down, but in case you're wondering how I know this is tan and brown, because there's two letters and you haven't seen genetics before, what you're always going to have is a dominant trait and a recessive trait. Now, the vast majority of the time, the capital letter is the dominant trait, and the lowercase is the recessive. But don't assume that on the ACT. I have seen them flip that before, and they've said so in the text when they introduce what these traits are. They've said something like the lowercase is the capital, or lowercase is the dominant trait. So be really careful. I've only seen them do it once, but the fact that they did it bothered me. It seems like a trick, but that also means they might do it again. So just watch out. So in that case, if you have even one of the dominant traits, that's the one that's going to win. So in this case where I have a dominant and a recessive, dominant being capital A, the tan, that means it's going to be the dominant trait, so tan instead of black. If it was lowercase a, then it would be black. It's the only way you can get the recessive is if the entire thing, so both of them, are the lowercase or the recessive gene. So what do I have to know about genetics? Well, first of all, in most cases, every trait's going to be determined by a letter with two alleles. So in this case, like I said, I have the fur color. So there was our being controlled by just A, the letter A. So it's not A and B controlling the fur color. That's what I meant by each trait's determined by a letter. And then that letter has two alleles. So I have two A's. So in this case, like capital A, lowercase a. So that's how that is determined. I have seen one of the more complicated examples where they actually had the combination of all four, so two genes working together to just give like a fur color. So in that case, you had a little bit more of a puzzle to try and figure out, well, what are all the combinations to give me all these different color choices? So if you do see that, just take your time and try to figure out which ones are dominant and how do these interact. but that is rare, so I'm not going to focus on that because I've only seen it once. I've seen a lot of the typical two alleles, so one letter for one trait, another letter for another trait. Next thing you have to know, if at least one of the two alleles is the dominant, as I was saying up here with the A example, so that's usually the capital letter, then the dominant trait is displayed. Whereas for recessive, both alleles, or all of the alleles, need to be recessive, so the lowercase. But again, don't just assume capital is dominant and lowercase is recessive. Most of the time that will be right, but double check to make sure that makes sense with what you're seeing on your chart, your graph, or what they're describing in case they flip it on you. The next thing to know is when these two dogs have a kid or child, each of those parents are going to donate one allele for each trait. So that means from the A's, dog 1 here is going to give either this A or this A. And then dog 2 is going to give this A or this A. And then the same will be true for the B's. And you combine them to get the child. So, since a recessive requires that both of the alleles has to be recessive, then if you want to have a recessive trait displayed, then both parents need to at least have one recessive allele, because it needs to get one from both parents. Whereas if this was capital A, and then this was lowercase a, despite the fact that this is displaying the recessive, all of the children of these two would then have the dominant trait, because they would all have capital A, lowercase a. And it would be impossible to have lowercase a. But in this case, that's not true, because there is at least one lowercase in each of these. So let's look at an example of actually getting the offspring here. And the way to do it is just focus on one trait at a time, and they're only going to ask about one at a time. So don't go and figure out all the combinations of everything unless they're interacting in that one very rare case I was talking about. So looking at the A's for fur color, here are the possible combinations. And you'll notice that it's 75% tan and 25% black. So 3 of the 4 are going to give you tan. That's because that's the dominant. And only one of them is going to give you black fur, because that's the recessive in this case. So notice again, even though here we have a recessive allele, it's still going to display the dominant trait because there's a one dominant letter. So if you're wondering how did I get these combinations, there's 2 ways you can do this. Let me get the pen. The way I prefer is to just draw lines for each combination. So let's start with this A right here, the first A. So if we assume we're getting the capital A from dog 1, well, I can draw a line over to each of the A's here. I can do this and get A and A if I just get the first of each. So that's where this one comes from, right there. Well, let me erase that one real quick, because that I just crossed out of it instead of underlining accidentally. So that's how I get this one. Now, if I come back to this first A, let's say I did not get this first A of dog 2, I could instead get this. So now I've drawn a line from this first A to each of the possible combinations on this side, and that's how I would get this one. Now, let's go back to dog 1 and say, well, what happens if I don't get the first A from dog 1? Then it would have to be the second A. So I would do the same thing. I would draw a line from that to each of the A's on the other dog. So that's how I could get lowercase a, capital A, which is, again, this one. I rearranged it because I always like to put the dominant first, because that's the one that's going to be displayed. But if I had lowercase a, capital case A, it would be the same thing. And just like before, there are 2 different combinations using that lowercase a. So it could come all the way over to that, where you could get lowercase a. And that's how I find these. The other way you could do this is with a chart. A Punnett square is what this is called. If you wanted, you could take each dog, so let's here, dog 1, and put it on one side, and dog 2, put it on the other side. And then you just look at each of these combinations. So in this column here, I'm going to get the capital A from the top, and then here in each column, I'm going to get the lowercase a. from the top. And now let's go across the rows. I'll get a capital A, and then from this row, I'll get a lowercase a to each. And that's how you can also get those 4 combinations. Now, let's erase that real quick. And move on to eye color, where we'll do the same thing. So this time, before I start writing all of the different combos, let's figure it out ourselves. So let's do both methods, looking at B. So we'll start with the first B in dog 1, and just draw a combo to each that we could have. So you can see I could have big B, or big B, little b. Those are both my options there. Now if I was to go with the second B of dog 1, in both cases, I would end up with little b. And that's all I would get here. Now if I wanted to do this with the Punnett square, let's draw this real quick. Excuse my terrible handwriting. Let's put dog 1 up top, dog 2 on the side, do columns. So I get the 2 B's, or capital B's, straight down the column there. Now let's do this next column where I'm going to get little b. Now let's go across the rows, where from this, I'm going to move these to the right and carry it out, so I'll get the little b to both of them. And we'll do the same thing on the bottom row. And that's how we can get all of our combinations to end up with these. And you'll notice here, since this one was actually recessive, I end up with 50% of them being brown and 50% blue. So even if one of them is completely recessive, both of them being lowercase, and there's a lowercase there, don't do the math of thinking, oh, look, of the 4, 3 of them are lowercase, therefore 75% of the children would have recessive, right? Not true. Because of the way recessive works, it actually turns out to only be 50-50. So make sure if they ask for a percentage like that, you look and do the math by drawing out your combinations or drawing out your Punnett square so you can figure out what actually would I get. So that's all you have to know about that, with the exception of this right here, where we just need to know, how do I read a pedigree chart? It's the last bit of genetics. So let me run you through this real quick on how to read this. So each of these, let's say these are people. Each square or circle is a person. And there's going to be a key somewhere telling you what they all are. So in this case, square means male, circle means female. And now you'll notice some are solid, some are empty. So it'll also be in the key saying, hey, if it's solid, some trait is being displayed. If it's empty, the trait is not being displayed. So they might ask you to use this fact to try and figure out, well, which one's dominant, which one's recessive. And you might have to try and figure out, well, something like, well, look at this one. In this case, both of them are not displaying this, and everything is not displaying. So this trait here that's not being displayed, the other one, this one might be the recessive. So who knows? In most cases, they'd probably have more information or more children to make it more obvious rather than just 2. But that's how you would read this. Each row would be another generation. So this is generation 1, this is generation 2, this is generation 3. If the 2 people have a child, you'll draw a line between them. So these 2 have a child, these 2 have a child, and these 2 have a child. Notice these 2 are next to each other, but they do not have a child because there's no line between them. They're connected like this. When you do have that child, they're connected, and then you draw a line down, and then you'll have a bracket like this for all of the children that this pairing had. So you can see this pairing had 3 children because it draws down and then it brackets down to 1, 2, 3. So these 3 are the children of these 2. So these would then be siblings. Notice that there's also these 2 on this row, but they are not the children of these 2 because this bracket did not extend out this far. Instead, these 2 would be spouses. So this person married or a partner that then I had a kid with this person. And then over here, this person had a kid with this person. So not necessarily related to these people, but they do enter in at this generation to produce generation 3. So then looking at that, generation 1, just to summarize, has 3 children and 3 grandchildren because their children had 1, 2, 3 grandchildren. So that's all you'd have to know about reading that. They might ask you questions to try and figure out which is recessive or dominant. That's less common. They might ask you, okay, how's this person and this person related? In which case it would be grandchild, grandparent. Or how are these two related? Parent, child. How about these two related? These are married or partners. These two would be siblings. So you just have to know how each of these people is related and figure out what's going on with the genetics. And that's all you have to know about how to read these pedigree charts. Now, while we're here, this isn't tested too often, but it's one of the more common vocab type questions in science, and that's the homo and hetero prefix. All you have to know is homo means same and hetero means different. So in the case of genetics, you're often going to see something like homozygous and heterozygous. Notice that both of these are the same, they're both capital, so homozygous. Here it's one capital, one lowercase, heterozygous. So same, different. You don't have to remember the zygous part, just homo and hetero will be able to figure out for anything where you see that. All right, next up, because since we're done with genetics, we're going to go to charge, current, and magnetism. These are very complex concepts that you're typically going to cover in a physics course that almost no students have gotten to when they get to the ACT, and they might not cover it at all in high school. But don't panic, because a lot of students will panic when they see this, like I've never seen magnetism, how am I supposed to do this? And they'll miss a lot of questions. Instead, focus on what you already know, which is the fact that they're not testing your scientific knowledge. That is in there just to try and trip you up. They're just trying to see, can you read the graphs? Because most of the time with these passages that have these, they're not going to ask much, if anything, about actual charge, current, and magnetism science. They're just going to ask you to read those charts and read those tables, just like any other passage. So don't panic. Treat it just like you would anything else. There are only 2 simple things that they might test about the actual science of these things. And that's it, just 2. And they're very easy once you know how to apply them. The rest of those questions are just going to be reading graphs, reading tables. So what are those 2 things? 1, like charges repel, opposite charges attract. So if I have 2 positive charges, those are like charges, so same charges. They're going to move away from each other or have forces pushing them in opposite directions. If I have a positive and a negative charge, they're going to be opposite charges. Then they're going to attract to each other. So they're going to be pulled together, move towards each other, or have forces experienced towards each other. That's rule 1. Rule 2 is that voltage, V, or electric potential, those are going to be used to accelerate or move particles, usually electrons or something. So if that voltage or electric potential is set to 0, that typically means that it's off. So you're not going to have a current, you're not going to have a beam of particles. And I'll show you on the next slide an example of what that will look like. I don't know if it's actually the next slide or the following, but I'll go through how you'll see each of these 2 things applied most of the time. So first of all, the like charges repel, opposite charges attract. You'll have 2 charges, and in this case, a positive and a negative. They're opposite. They're going to move towards each other or feel forces towards each other. In this case, they're the same charge, so they're going to move away from each other or feel forces away from each other. You might see this, instead of 2 just plain charges, you might have 2 wires that have charges. In this case, if these were wires, they would bend towards each other, and in this case, they would bend away from each other. But again, it's the same rule of like charges repel, opposite charges attract. You'll also sometimes see a beam going through a plate, and this is the more complicated one that people get really confused, but once you understand what's happening, it's really simple. Where we're going to have some beam that has a charge. It might be a positive charge, it might be a negative charge. They might have something like this saying, hey, it's positive, or you might have to read in the text to say what charge does this beam have, or laser. Then there's going to be 2 charged plates on either side that's going through, and they will have some charge, usually opposites. So in this case, I put the top one as a negative charge, and the bottom plate will have a positive charge. and we're going to shoot this beam through these plates. And what's going to happen is it's going to bend the beam, and they might ask you to know which way it's going to bend. And you might think, how on earth would I know? But it's just following the same rule of like charges repel and opposite charges attract. So let's take one plate at a time. If I have a positive beam and a negative plate, those are opposite charges, so they're going to attract. So this beam is going to be pulled towards this top plate, which is why it's bending upwards. Same can be true for the positive charge on the bottom plate. They are now the same or like charges, so they will repel and push away from each other. So this beam is going to be pushed away from the positive, making it bend upwards again. So that is why it bends this way. And in such an experiment, they'll maybe have some kind of meter or paper or something here that's going to measure where does this beam hit. And they might change things like how strong is this getting pushed, the velocity of these, or how strong are these charges to see how much does it bend and where does it hit. Does it hit up here, 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 down here, that sort of a thing. And so that's all you have to know about which way is it going to bend. And then you also have to know, as I was saying before, that voltage V or electric potential are going to move those particles. So if you don't have that, in the case of a current, it's just going to be off. Or in the case of a beam like we just saw, there's going to be no beam of particles. So if I go back to here, that current or electric potential of something over here is what's going to shoot this beam out. And then we're going to measure where the beam hits. If that current or electric potential is 0, it can't shoot the beam. So there's just going to be no beam, and it's not going to measure anything over here. It's that simple. So in that case, the answer, like, well, if, because I have seen an exact question where like, okay, what happened when it measured that the beam hit at 0? So it didn't bend at all. And one answer was that the voltage of this thing was set to 0. Another was that there was no charge on the plates or no charge on the laser particles or whatever. And the answer there is that there has to be no charge. Because if this was 0, the voltage or electric potential, it wouldn't have a beam at all, so it never would have picked up anything on this paper at the end. Whereas if it does have a voltage or potential, it still shoots the beam, but if there's no charges, nothing's going to bend it, so it would just go straight through and hit dead center. And that's all there is to it. They seem really complicated. This diagram looks really complicated, but just take a second, go look over these slides again, understand those two concepts and how to apply them, and you'll be good to go. That's all you're going to need to know for the ACT. The rest, again, is just going to be reading the graphs and reading the tables. So with that, good luck, and I'll see you on the next one.